You've probably heard that phrase before. Cancer runs in the family. It's a really chilling thought, right? This idea that a hidden danger could be passed down through generations. But for scientists and for these families, that shared risk is often this profound and deeply personal mystery. So today, we're going to dive into how they're starting to crack that code.
I mean, that's the big question, isn't it? For so many families who live under the shadow of a hereditary disease, the answer just feels frustratingly out of reach. They see the pattern, this terrible inheritance, but they have no idea why. Well, today, we're going to follow the genetic detectives who are trying to find those answers. So let's zoom in on one specific family story. This is a family with a painful history of colorectal cancer, and they were absolutely determined to uncover the genetic root cause that had affected so many of their loved ones. And they weren't just looking for an answer for themselves, but for a clue that could end up helping everyone.
Okay, so this is where the investigation starts. This is the first piece of evidence, a pedigree chart. It's basically a family tree for geneticists. The squares are males, the circles are females. And those colored in shapes, those show the family members who were diagnosed with colorectal cancer or precancerous polyps. The pattern is crystal clear. The risk is being inherited. But the why, the specific genetic flaw causing it, is a total unknown.
And this right here is why this case is so important. This family situation, it's not unique at all. For the vast majority of families dealing with this type of cancer, we're talking a whopping 90 to 95% of them. The usual genetic suspects, the well-understood gene mutations, they just aren't there. The cause is a complete mystery, buried somewhere deep in our DNA.
So how on earth do you even begin to look for a single typo in a genetic library that has billions of letters? Well, the investigators started by using this incredibly powerful tool to scan the DNA of family members, both those who were sick and those who were healthy, just looking for any clue, anything that stood out.
That tool is called whole exome sequencing, or WES for short. Okay, think of your entire genome as a massive library filled with thousands of instruction books. Instead of reading every single word on every page, which would take forever, WES is a shortcut. It focuses only on the most important parts, the exome, which contains the actual instructions for building all the proteins in your body. It's a really smart way to hunt for clues.
And the first scan came back with this. Over 13,000 potential genetic suspects or variants. I mean, talk about a needle in a haystack. This is a haystack of monumental proportions. Figuring out which one of these is the single culprit, that's the real challenge.
So how did they do it? Well, they used a process that works kind of like a funnel. They started with that huge list of over 13,000 variants. First, they filtered it down to only the ones shared by the family members who actually had cancer. Boom, down to 783. Then, they used some really sophisticated analyses to whittle that list down and down and down until only 2 prime suspects were left standing.
All right, let's meet the first major lead in this genetic detective story. It was a variant in a gene called APCDD1, and on paper, it looked like the perfect culprit. Honestly, it was exactly the kind of clue that researchers expect to find.
So here they are, our 2 finalists. On one hand, you have the APCDD1 variant. This was the obvious suspect, right? It was found right in the main text of a gene, the part that actually gives the instructions. And then there's the other one, a variant near a gene called HDAC5. This one was different. It was in the non-coding DNA, the part that, for years, scientists kind of dismissed as junk. This was the hidden clue.
And listen, the case against APCDD1 was strong. It was in a protein-coding gene involved in something called the Wnt signaling pathway, which is a super-critical process that controls how our cells grow. And in colorectal cancer, this pathway often goes completely haywire. Finding a mutation here, it was like finding a suspect at the scene of the crime with a very clear motive. It looked like an open and shut case.
But, as with any good detective story, just when you think you've got it all figured out, there's a twist. And the evidence that looked so perfect, it was about to completely fall apart. When the researchers took this APCDD1 variant into the lab to test it, the results were a huge shock. The variant had an insignificant proliferative impact. In plain English, it didn't actually cause the out-of-control cell growth you see in cancer. The prime suspect had a rock-solid alibi.
So it was a complete dead end. All that work, all that incredible filtering, and it led them to the wrong conclusion.
The genetic detectives had to go all the way back to the evidence board and take a much closer look at that second, much more mysterious clue, the one they had almost overlooked.
So now, the whole investigation pivots to this HDC5 variant. This clue was located in the so-called junk DNA, those vast non-coding regions of the genome that don't make proteins, but which, as scientists are now realizing, are anything but junk.
You know, this discovery is reframing our entire understanding of DNA. For decades, we've focused on just the 2% of our genome that makes up our genes, but the other 98%, this non-coding DNA, it's not junk at all. It's more like the body's operating system, a massive, complex switchboard that's constantly telling our genes when to turn on, when to turn off, and how loud to be.
Now, this specific variant was found in a regulatory region right next to the HDC5 gene. So, going back to our library analogy, this isn't a typo in the main story. It's like a tiny mistake in the footnotes at the very beginning of a chapter. And as it turns out, that one tiny mistake can change the meaning of everything that comes after it. And here it is, the breakthrough. When researchers tested this variant in the lab, they got a totally different result. It led to enhanced promoter activity. Basically, this tiny variant was acting like a faulty light switch that was stuck in the on position, cranking up the activity of that nearby HDC5 gene. And that's a problem, because HDC5 is a master regulator that's supposed to shut other genes down. So too much of it is like a power surge that turns off the very genes meant to prevent tumors. They had finally found their culprit. This discovery wasn't just a victory for this one family. It was a landmark moment that points to a much, much bigger picture. And it's changing how we think about hunting for the causes of hereditary diseases altogether. I mean, this visual really says it all, doesn't it? For so long, we focused all of our attention on that tiny 2% slice, the genes that tell our bodies what to build. But this case proves that the secrets are often hidden in the massive 98% that tells our bodies how and when to build it. So, this is the big takeaway. The solution to countless genetic mysteries, from cancer to heart disease to all sorts of other disorders, it might not be in the genes themselves at all. It might be hiding in plain sight, right there in the vast, complex regulatory code that we're only just beginning to understand. Which leaves us with this final, really fascinating question. This family's incredibly personal search for answers led to a profound discovery in the so-called junk of our genome. It really makes you wonder, what other secrets, what other cures are still just waiting to be found in the 98%?